cell spends the majority of its time in the cell cycle and interphase. This is where it practices daily functions, growth, and prepares for meiosis and cytokinesis. It is split into 3 stages. The first is G1. Next is synthesis, where chromosomes are replicated and DNA is synthesized. Finally, in the G2 phase, the cell continues to prepare and grow. All in all, interphase takes up 90% of a cell's time in the cell cycle. Let's now move on to meiosis 1. The first phase is prophase 1. In prophase 1, the chromosomes condense to form homologous pairs, and the 2 homologous pairs form a tetrad. The chromosomes also begin crossing over to exchange genetic information. After prophase 1, we move on to metaphase 1. In this phase, the tetrads move towards the equator and line up in a direct line down the middle. The spindle fibers then begin to form and attach to both sides of each tetrad, including the centromere. Up next is anaphase 1. During anaphase 1, the tetrads separate and homologous chromosomes move towards opposite poles. Finally, we move on to telophase 1 and cytokinesis. In telophase 1, 2 new nuclear membranes reform around the separated chromosomes. While in cytokinesis, the 2 new haploid cells separate by a formed cleavage furrow. We are now in meiosis 2, beginning with 2 haploid cells. This begins with prophase 2. In prophase 2, 3 important steps happen in the 2 genetically unidentical haploid cells. First, the chromatin condenses into chromosomes. Next, the nuclear envelope breaks down. Finally, the spindle begins to form. We then move on to metaphase 2. Similar to metaphase and mitosis, the replicated singular chromosomes line up in the middle. The next phase is anaphase 2. In this phase, the spindle fibers pull the sister chromatids apart and towards opposite poles, just like in mitosis. This forms non-replicated chromosomes. To finish off meiosis, we arrive at telophase 2 and cytokinesis. In telophase 2, the 2 haploid cells begin to divide and nuclear membranes form. In cytokinesis, the cleavage furrow forms and separates these into 4 genetically different haploid daughter cells. This random assortment causes genetic variety, and these unidentical 4 cells are the final product of meiosis. This is how all 7.5 billion people in the world are unique in their own way. In this photo, it shows chromosomes from the mother, which are red, and green chromosomes from the father. When a baby is produced, meiosis occurs, causing a baby to be unique genetically but similar to their parents. I will now talk about diploid versus haploid cells. Diploid cells contain 2 complete sets of chromosomes, which explains the 2N. They also reproduce to create genetically identical cells through mitosis. Examples may include skin or muscle cells. Meanwhile, a haploid cell has only 1 complete set of chromosomes. This is half a diploid's amount and is easier to remember as both hap and haploid start with an H. Haploid cells are a result of mitosis, and examples include gametes, which are sperm and egg cells. Thank you so much for watching our video.